All right, so thank you very much for coming, and thank you very much for Ethan, the OAP member, the PR member, to give me a present to present my research on my birthday. Actually, today is my birthday. Thank you. Okay, so I like to start I like to talk about my research, my life as a microchip. So nowadays, there is no doubt about the importance of drugs. So when we got fever or stomach aches or some diseases, so we need to have drugs to cure those symptoms. And there's a huge market for the drugs now. But however, for the pharmaceutical company, they face a big problem to develop just one drug. For example, the time. So over the 10 years, and the cost over that 100 billion yen to develop just one drug. More seriously, the third problem is failure at the clinical trials or side effect for the patient. So those problems are quite serious issue for all the patient and the patient's family and pharmaceutical companies. So of course there's a number of reasons, but I like to explain about it one by one. And here is one example. So this compound. So do you know this compound? Can you guess what it is? How about the chemist? Okay, so this compound has a really long name as shown here. But this compound has the other names, like rofecoxib or Vioxx, which is the non-steroidal anti-inflammatory drugs, NSAID, or one of the most famous drugs in the world. So I'd like to tell you about this story. So Vioxx was originally commercialized from Merck for curing pain, osteoarthritis, and dysmenorrhea. So after getting into the market, so there's a number of reports coming up to suggest that the Vioxx caused to the heart attack and stroke with a long-term and high-dose usage. That's a quite serious problem. So after long discussion, so finally, Merck decided to recall this drug. So here is a stock chart of Merck in 2004. So as you can see, there's a big drop. And this is exactly when Merck decided to recall this Vioxx and lost $5 billion US immediately. So of course, there's a number of reasons behind. But I like to point it out the one thing, the preclinical trials with such cute animals. So basically, the preclinical trials were conducted for test the drug safety, toxicity, and some pharmacological tests. However, of course, the animal models are totally different from people's. As you might know, maybe. So there's a big gap behind. I hope. So even if we confirm that some drugs are very safe with the animal model, but some drugs might cause some serious toxicity in the patient. So by using animal model, so we cannot predict such a toxicity or side effect. So moreover, for the use of animal models, so due to the ethical issues, we need to follow 3 R's, like replacement, reduction, and refinement. So because we need to use a large amount of animals, but due to such an issue, we need to try to reduce, and we need to try to follow such a 3 R's. So on the other hand, so instead of animal models, we can also test a drug with biochemical and cell-based assay with microtitrat plate, as shown here. But it's quite obvious that these microtitrat plate conditions are totally different from the condition in our body. The main difference is the circulatory system. So each tissue's organs in our body are connected through the circulatory system. And drugs also travel through the circulatory system and influence to each organ differently. But by using microtiter plate, we cannot test such an experiment. Moreover, for the preclinical trials, we also need to consider about the ADME, absorption, distribution, metabolism, and excretion. So those are very important parameters to test for the drugs. However, using a microtiter plate, we cannot test such a drug. So we need to have an alternative way to test such a ADME. So here, I'd like to introduce our approach, body on a chip, to mimic the human physiological condition in a microchip. And actually, so I have here, this is a body on a chip, it's a prototype. And I'd like to introduce this one step by step. So to develop the body on a chip, there's two key elements. The one is iPS cell. The other is microfluidics. So moving forward to the iPS cells, induce pluripotent stem cells. So iPS cells, so we can obtain by using somatic cells to introduce a reprogramming factor. So these iPS cells has two distinct, very important characteristics. The one is unlimited self-renewal. The second is differentiation capability into almost any kind of cell in the body. So this means that by using iPS cells, we can obtain sufficient amount of tissue collection for further testing, like drug screening. Okay, moving forward to the microfluidics. So microfluidics has an advantage to control the small size of liquid, like water, from the nanoliter scale. So if you think about your body, actually our body consists of 60% of water, and this water plays an important role, like circulatory system, as I'm showing here again. So actually, as you can see here, you can realize that our body has a very complicated fluidic system. So, but by using microtiter plate, we cannot mimic such a fluidic system. So microfluidics is coming to play. So we are microengineer. So we can establish micropipe, pump, valve into the microchip and establish a very complicated fluidic system, as shown here. Actually, this is my colleague established a very complicated fluidic system, like subway map in the Tokyo station. So by using this one, so we can make a circular system, go through and stop by. So we can do many kind of complicated stuff. So we have a capability to establish the artificial circulatory system in a small device, as I shown here. Moreover, as I mentioned, we are molecular architect. So we try to build very comfortable house for cells. Actually, comfortability is very important for the cells. So if you think about yourself, so if you live in very uncomfortable house, and then if you cannot sleep well during the night, so you cannot work during the day. So that's a problem. And these things happen for the cell as well. So like a microtiter plate, we cannot get cell's own function. Therefore, we need to build such a comfortable house for cells. So our approach to develop such a body in a chip as follows. So we prepare the microfluidic device which has the comfortable house for the cells and circulatory system. Also, we prepare the microtissues from the iPS cells as much as possible. Then introduce into the microfluidic device. Then we apply the circulatory flow with drugs to test and to see what's going on for the ADME, like absorption, distribution, metabolism, and excretion. So this is phenomenon. It's totally different from other conventional techniques. So this is our body on a chip approach. So because of the use of iPS cells, we can also establish the personalized and disease-specific body on chip. So as you might know that some drugs affecting each patient differently. So it's a kind of the personalized difference. But the conventional method we cannot test such a difference. However, if we can obtain the iPS cell from each person, so we can also test the difference of those drug efficacy by using our body-on-chip technology. So this means that if we can create the iPS cells from all the members of you, iCEMs, then if we can put it into the device, we can generate the iCEMs on the chip. So this is one of my dreams. Okay, so thank you very much for your kind attention. But I do have one more thing. Yeah. So I introduced body on a chip, but is it only for the people? Actually, not really, right? So if we can generate the iPS cells from the animals, we can also generate the body on a chip for the animals. So we try to explore the more possibility of the body on a chip for further testing. So we are aiming to secure endangered species. So nowadays, there are over 3,000 of endangered animals on Earth, and they are very facing the problem for their survival. So nowadays, the modern zoo are getting very important role for the conservation of such animals, endangered animals. But if those animals get sick, they cannot cure those animals because they don't have any effective medicine or drugs to cure those animals. So the one big reason is that we don't have any research tool to study about their diseases because it's so limited. So our approach to tackle these issues as follows. So if we can generate, actually we can obtain the somatic cells from the endangered animals here, non-invasively, and then we can establish iPS cells out of that. Afterward, we can establish tissue collection from the iPS cells and then put them into the microfluidics to generate the gorilla, chimpanzee, or whatever which we need for the body-on-chip technology. So as you may realize that, so we are not aiming for, we are aiming not only for the peoples, we are also aiming for wildlife conservation. Since as a member on Earth, I like to contribute for the global health care of all the animals living on Earth. Okay, thank you very much for your kind attention. Thank you very much.